Hello everyone and welcome to our channel Applied Forensic Research Sciences. Myself Dr. Pankti Patel, MSc Forensic Odontology and volunteer at AFRS. So let's talk about Introduction to Human Osteology Part 2. The contents of the presentation include first, structure of bone, second, bone cells, and third, development and ossification of bone. So let's start with the structure of bone. Macroscopic structure. It is divided into first, compact bone and second, cancellous bone. Compact bone is hard and dense and it contains no bone marrow space. Cancellous bone is spongy, trabecular framework which is arranged in definite pattern which resists the stress, and it contains bone marrow. So as you can see in the diagram, this is structure of a compact bone. The ends of the bone are known as proximal and distal epiphysis, and the middle portion is known as diaphysis or the shaft. There are two layers of the bone. The outermost layer is known as the periosteum and the innermost layer is known as the endosteum. And within this runs a medullary cavity in which the yellow bone marrow is found. Along with that, there are numerous nutrient arteries, vessels and veins which run along the surface of the bone. Coming to the microscopic structure, bone is made up of numerous cylindrical units called as the Haversian system or osteon. The Haversian system consists of four main elements. First, the Haversian canal, second, lacunae, third, lamella, and fourth, canaliculi. Haversian canal. It is the canal present in the center of system. It runs parallel to the long axis of bone. Each canal contains artery, vein, nerve and lymphatics with supporting areolar tissue. Lamella. Lamella is a thin bone tissue containing ground substance or matrix or collagen fibers which are arranged concentrically around the canal. They are interconnected with other fibers. Lamella are of two types. First, interstitial lamina. They lie at angular interval between the Haversian system and they run in various directions. Second, circumferential lamina. They are found over inner and outer periphery of cortex. Lacuna. Spaces between the lamella are known as lacuna. Each of these contain a bone cell. Canaliculi. They are fine radiating channels that connect the lacuna with central Haversial canal, and they contain the protoplasmic processes of the bone cells. This is a diagrammatic representation which explains the two layers. The periosteum contains outer fibrous and the inner cellular layers. Collagen fibers of the periosteum are continuous with those of the bone, adjacent joint capsules, and are attached to tendons and ligaments. Second is the endosteum. Endosteum is an incomplete cellular layer containing osteoblast, osteogenic cells, and the osteoclast. Bone cells. Mainly, there are three types of bone cells, all of which are derived from osteogenic cells but have different functions. First, osteoblast. They are ovoid with basophilic cytoplasm responsible for active bone formation. They form protein of matrix and control mineralization. They contain alkaline phosphatase. Second, osteocyte. Osteoblasts change into osteocyte by developing matrix around itself, become dormant when active bone formation is not required, and they will occupy the spaces in lacuna. Third, osteoclasts. They are large multinucleated cells with acidophilic cytoplasm, responsible for bone resorption and bone remodeling, contain acid phosphatase. This figure shows the anatomic structure of three types of bone cells. So as you can see, osteoblasts are ovoid with single nuclei. Osteocytes have a main cell body along with the radiating fibers around the periphery. Osteoclasts are multinucleated with a rough and ruffled border or periphery. Now coming to the last section of the presentation, development and ossification of bone. Bones are mesodermal in origin. They develop from the embryogenic mesenchyme, which is a loose connective tissue with fluid matrix. Basically, bone formation is done in two types, endochondral or intramembranous. Endochondral. Here, the bone develops by replacing cartilage. This is seen in almost all bones of the skeleton like the vertebrae, pelvis, long irregular bones, the base of the skull except for clavicle. Intramembranous. Here, bone develops from fibrous membrane. This is seen in the growth of short bones and the thickening of long bones. Intramembranous bone formation is seen in the cranial and skull bones, maxilla, mandible, flat bones and clavicle. This diagram explains the stages of bone formation. Stages of endochondral ossification. First, differentiation of chondrocytes, which are the cartilage forming cells. Second, formation of cartilage model of future bony skeleton. Third, periosteum formation with periosteal collar development, and there will be primary ossification center which will start developing. Next, the cartilage and the chondrocytes will continue to grow out at the ends of the bone and secondary ossification center will develop. Lastly, the epiphysis will ossify and the cartilage will remain at the epiphyseal growth plate and joint surfaces. Now the stages of intramembranous ossification. First, there will be ossification center appearance within the fibrous connective tissue membrane. Next, the bone matrix will be secreted, which will be later mineralized within few days. Woven bone and periosteum will form, and lastly, the bone collar of compact bone will form and bone marrow will appear.
Thank you so much. Do like, share, comment and subscribe to our channel Applied Forensic Research Sciences for more such sessions and interesting topics. In case of any doubts, you can post your questions in the comment section below.